And so our next presenter is Dr. Jonathan Casey, who is a clinical researcher and assistant professor at the Department of Medicine, the Vanderbilt University Medical Center. So his research focuses on comparative effectiveness and pragmatic trials. And he's going to talk about REDCap as a tool for that's useful for adaptive trials, I believe, right?
You got it. All right, let me share my slides here and we'll get going.
Yes, please.
Great. Are you guys seeing those?
Yep. Thank you.
Great. So thanks again for the introduction. Again, I'm John Casey, Assistant Professor of Pulmonary and Critical Care at Vanderbilt University Medical Center. My funding is from the NHLBI, Department of Defense, PCORI, and NCATS, none of which are relevant to the talk today. Our agenda today, we're going to talk about platform adaptive and decentralized trials, quickly remind ourselves about what problems these trials are trying to solve. We'll review quickly some adaptive trial design types, and then we'll talk about the requirements of a randomization system for platform adaptive and decentralized trials. Then we'll turn to talk about REDCap, which is really what I'm here to talk about today, and its historic functionality to support randomization within a cloud-based electronic data capture system, the capability gaps of that system for platform adaptive and decentralized trials, and the design and release of REDCap Randomization 2.0. And then finally, we'll finish with what I think is the toughest question for adaptive trials. So I want to start with a disclaimer. So who am I? I'm a clinician, a pulmonary and critical care physician. I'm co-PI of the Vanderbilt Trial Innovation Network that you heard Dr. Hanley talk about earlier. And I'm the director of the Pragmatic Critical Care Research Group. What am I not? Unlike our other speakers today, I'm not a biostatistician and not an expert in adaptive trials or randomization. So I'm happy to turf all of those tough questions about many of the models back to our other speakers today. But I'm excited to talk about our work through my role in the Trial Innovation Network to build the REDCap tool to support these kinds of platform adaptive trials. So let's talk first about what a traditional randomized trial is. So a traditional randomized trial pre-specifies all trial procedures, including eligibility criteria, group allocation, intervention, and sample size. And those trial procedures proceed unchanged throughout trial conduct. So that's shown here in a little cartoon. Those types of trials may have one or many interim analyses. The trial might stop or not stop at those interim analyses, but nothing about learn from accrued patients changes the conduct of the subsequent trial procedures or allocation ratio or anything else about the trial. What are challenges of these traditional trial designs? Well, there are a lot. So a partial list is shown here. They're expensive and they take a long time. They're designed to answer only one question. They rely on assumptions regarding baseline event rates and outcome distribution and effect size. They commonly provide results that are uninformative, usually due to being underpowered. They may be affected by imbalances in key baseline characteristics. Patients may have concerns regarding being randomized to placebo, and many trials have problems with being unavailable to certain types of patients or patients from certain places, and therefore have poor generalizability. And for each of these challenges, there have been solutions proposed. Those include for the cost, duration of trials. People have proposed platform trials, trying to answer more than one question on the same platform. Using Bayesian analysis, specifically Bayesian sequential designs or sample size re-estimations and a frequentist framework to try to prevent trials from being underpowered. Using adaptive randomization to try to minimize imbalances in baseline characteristics, or adaptive randomization to change allocation ratio and address patient concerns regarding being randomized to placebo. And finally, decentralized trials and pragmatic trials have tried to broaden the availability of trials and address the generalizability and trial access issues. So not all of the proposed solutions we talked about are so-called adaptive trials, but I think it is helpful to remind ourselves what kinds of adaptive trials exist. And this is certainly a small and incomplete list, but I think might help when we're thinking about what kinds of tools we need to support these types of trials. So first, platform trials. Platform trials may or may not have any adaptive features, but commonly platform trials add or drop arms based on knowledge from prior participants, which is typically referred to as an adaptation. They may also randomize patients at multiple points in time. For example, randomizing patients to an intervention and then taking patients who fail and randomizing them to a rescue therapy, or they may randomize patients in multiple domains. And the goal with these types of platform designs are to answer multiple questions within a single trial. Bayesian sequential design, by comparison, is doing frequent interim analyses that try to continue the trial until it accrues definitive evidence of efficacy, harm, or futility. Adaptive randomization is taking knowledge gained from prior participants to either to change allocation probabilities for future patients, to either minimize baseline imbalances, so covariate adjusted randomization, or to try to increase randomization to better performing groups, so-called response adaptive randomization. And finally, you might use predictive enrichment. So look within your trial about which patient populations appear to be benefiting and then selectively target those patients in subsequent portions of the trial. So what would you need from a randomization system to support these kinds of platform, adaptive, and decentralized trials? Well, so a short list of tools that you might need would include the ability to add or drop groups, the ability to perform multiple randomizations in the same record, the ability to do adaptive randomization to generate randomization sequences that change allocation probabilities based on the characteristics of prior participants. As mentioned, that can either be based on covariate adjusted randomization to minimize differences in baseline characteristics, or outcomes of prior participants, response adaptive randomization. And then finally, decentralized trials, the ability to conduct randomization without the involvement of research personnel, like randomization in the back of an ambulance or randomization in a routine rural health clinic. Many of you are familiar with REDCap. It's one of the most commonly used electronic data capture platforms. It's a free, secure, web-based platform. It has over 7,541 active partners in 159 countries. It has over 2 million projects involving nearly 3.5 million users, and has been cited over 40,000 times in journal articles. And since REDCap was developed here at Vanderbilt by Paul Harris, it's included an embedded tool for randomization. So let's talk a little bit about that tool and what it has been able to do. So some screenshots from one of our trials are shown here and show how it's a pretty user-friendly tool that's embedded in a REDCap data instrument. It allows a single randomization for each study record. And using the logging function in REDCap, there's metadata created around this randomization so that we know who performed the randomization and the date and time at which it was performed, which is helpful in the case of audits or data queries. This randomization tool does a lot of the things you might want. So it allows you to stratify by site and to stratify by additional characteristics like age and sex. What can it not do? So historically, in this tool, once you set the randomization schedule, it was fixed and couldn't be modified for the duration of the trial. So that made it impossible to add or drop groups or change allocation ratios. You also could only randomize each patient once per study record. So you couldn't do a platform trial that would randomize into multiple domains or at multiple points in time. It had limited ability within the embedded randomization tool to link to external software to do the kinds of advanced modeling that are required for adaptive randomization and to query other participants' baseline characteristics or outcomes. And then further, randomization could only be conducted by study personnel with access to the database. So you couldn't embed it in a survey and use a QR code to access the randomization tool in the back of an ambulance, for example. And then finally, it required additional steps to blind users to group allocation. So it didn't have some of the protections you might want for a placebo-controlled trial, for example. So let's think about a hypothetical trial. And I'm, as mentioned, a pulmonary and critical care doctor, and a lot of the work I do is in the acute care space and emergency interventions. So this is a trial that I could think about conducting. So a placebo-controlled dose-finding trial of a novel antiarrhythmic following prolonged cardiac arrest. You could think in a trial like this, you might have thousands of paramedics who are caring for these patients. And if you need to start immediately, those are the people who would have to identify, enroll, randomize, and deliver the intervention for these patients. You'd have to be able to randomize in the back of an ambulance before any key study personnel are present. You'd have to blind clinicians. If it's placebo control, you need to blind clinicians, and you may want to use numeric identical numbered medication packs for tracking and auditing. If it's a dose-finding study, you may want to evaluate at the interim analyses which arms are performing worse and drop an arm when transitioning from a phase 2 to phase 3 trial, for example. And especially if it's a trial where the patient population is hard to find and you're enrolling at a large number of centers and may have a small number of patients per strata, you may be worried about imbalances in baseline characteristics and may want to use adaptive randomization to minimize those differences in baseline characteristics. And you may also want to continue the trial using a Bayesian sequential design until you have definitive evidence of efficacy, harm, and futility. So which of these trial features could be supported in the historical REDCap randomization tool? Well, as mentioned, you couldn't have the clinicians randomize patients. That you'd have to have all 3,000 or some odd paramedics in the trial registered as key study personnel, listed in the IRB applications, and have them have dedicated REDCap user accounts, which is really impractical. And because of that, how have studies like this been conducted? Well, historically, even up till today, most of these studies are either conducted at the cluster level, which has its own challenges, or using paper envelopes, which have been commonly misused even in recent pre-hospital studies. So I think this is a real need. We've been unable, there are some tools and ways to blind that we'll talk about within the historical tool, but they've been insufficient. And they specifically don't give you randomization numbers that you can match to med packs. There's been no way to change the allocation ratio, the allocations for future participants, so you couldn't drop or add arms or change allocation ratios. And I don't want to mean that these barriers have prevented any adaptive trial types, and there's certainly no reason that we couldn't have used the REDCap randomization tool and still done a Bayesian sequential design. In starting to explore this problem, we engaged a lot of statisticians and coordinating centers to see how they were dealing with this. And it was interesting to hear what workarounds people had developed. One was that people were copying their study databases just to get them back into draft mode and unlock these future randomization tables. So one of the stakeholders we engaged was copying their entire study base every week and porting over all their user data access rights just to be able to change the allocation for new patients that might be randomized in the following week. And that led to huge problems, including loss of metadata that might be problematic in the time of an audit. Further, some users had used API tokens to basically custom build their own randomization tools. So they abandoned REDCap's tool and custom built their own, including building their own, coding out their own randomization buttons. And many of these tools worked well, but they required really a lot of time and expertise from the person coding them. And they lost many of the data protections that were intended for the REDCap randomization tool, such as metadata. And then many users, not surprisingly, felt forced to abandon the REDCap randomization tool in favor of other cloud-based tools that allow them to do these kinds of adaptive randomizations. Part of our mandate in the Vanderbilt Trial Innovation Network has been to address this problem. So we convened a team to work on developing REDCap Randomization 2.0. The members of that team are shown here. It includes the next speaker, Frank Harris and Frank Harrell, and the founder of REDCap, Paul Harris. And I want to highlight in the top right there, Luke Stevens. He was a REDCap developer who volunteered to join the project. He works at the Murdoch Children's Research Institute in Australia and really showed himself to be an expert in this area and helped us to not only to identify the needs, but he had already developed a module that addressed many of these needs and helped us code out some of the fixes, and some of the slides that I'm going to show in the coming section are his. So great thanks to Luke. In terms of the timeline, we started working on this problem about a year ago and formed that working group. We, after framing out the problem, did a design studio where we engaged stakeholders, biostatisticians, and members of coordinating centers. So thanks to anyone in the audience who joined that or contributed to this discussion. We finalized the list of our needs by this spring and started building the new REDCap randomization tool, which was completed in September. We then solicited volunteers to help test the REDCap randomization tool, made some final adjustments, and then I'm happy to announce, if people haven't seen already, that this was released just last week. So it was released on October 9th via REDCap version 14.7. So what tools did we develop and build into REDCap Randomization 2.0? So those are listed here. They include multiple randomization, improved support for blinding, additional metadata for randomization, support for decentralized trials, ability for administrators to edit allocations, easier connection to API tools and hook functions. So we're going to go through each of these in a little more detail, and this is the section where I have blatantly stolen Luke's slides that he presented at REDCap Con talking about the new tool. So thanks again to Luke. So first, multiple randomization. So I think this is the basic and most basic but most important change. So compared to the old tool, where once you set the randomization tables, they were fixed forever, you're now allowed to have multiple randomizations, which does two things. So you can, within the same record, have multiple randomizations so that in a platform trial, for example, you can randomize the same patient into multiple domains or at multiple points in time. And then also you can hide an old randomization and show a new one. So if you want to change the add or drop a group or change the allocation ratio for future participants, you can do that easily within the new tool while still having all the protections that are built into the REDCap randomization tool, still having your old randomization sequence, which shows the full initial randomization allocation and all the patients that were randomized in that using that tool, while still changing the allocation plan for future patients. So this is a pretty basic change, but a very important one. Blinding is the next thing we addressed. So as mentioned, the prior REDCap tool didn't have built-in support for blinding. So by default, when you click that randomization button, it revealed group assignment directly. You could use some other tools that weren't built for the randomization tool to try to create a blinded randomization. So for example, you could have them click the randomization button, but then hide the field that came after it and use branching logic to try to show some coded allocation. But that was a system that was error prone, that could lead to unintentional blinding, and it didn't have support for randomization numbers. So in many other systems, people like not just knowing what the group allocation might be or using a blinded terms, but having specific blinded randomization numbers that can ensure blinding and ensure that only unblinded investigators, like the unblinded pharmacist, has the ability to look up what whether that randomization number is a placebo or an intervention. And then also in many trials, they use numbered medication packs where the randomization number matches a medication pack in pharmacy, and having that randomization number in the study record improves tracking mechanisms and allows for better audit transparency. So in REDCap Randomization 2.0, we now have the ability to allow the person designing the tool to reveal for the end users either group allocation or randomization number. And you can see here at the top right how you choose which one you're going to use and which field you're going to put that into. So when using a randomization number, when blinding the person performing randomization, an unblinded personnel can translate that randomization number to either to a numbered medication pack, or if they are using a lookup table, they can look up in a table that only they have access to, whether that randomization number corresponds to drug or placebo, or we can use other tools in REDCap that will pipe that information to a separate database. So only the unblinded investigators will have access to the group assignment, and it lives behind a firewall of a separate project, but all lives within REDCap. So this is all new functionality that will really help support these blinded trials. We've also improved functionality for metadata in REDCap Randomization 2.0. So I mentioned this as being a strength of the prior model, that we could tell who performed randomization and the time they performed it. But that information really lived within the metadata of the database and wasn't itself data elements. So when you exported data for analysis or for audits, that information was hard to export and hard to access. So it existed, but it wasn't easy to include in your study data set. And we've solved that problem the way that we have for many surveys, which is using something called smart variables. So these smart variables automatically calculate or take things from other places in the database and can either show them to the investigator or hide them, but have them available at the time of export for the biostatisticians. So that now it's very easy to build into your data set things like the date and time of randomization, either in the time zone that randomization is performed or at universal time, to show both the randomization number and group allocation. So all those things are now allow for better tracking in terms of audits at other times. Next is one that I think is really important for my work, which is the support for decentralized trials. So as someone who works a lot in the emergency care space and still is stuck using paper envelopes, I've been excited about the idea of using this REDCap randomization tool and embedding it among clinicians who perform randomization in many of our trials. So REDCap Randomization 2.0 now allows that. So the way it does that is by performing randomization when a series of logic steps are true. So you can think about the way this plays out is that you could have a clinician use a QR code, and it will pull up a survey, and then they'd be required to answer a series of questions like the study, the site, the patient's name, their name, and then when they submit that survey, this tool will fire, and if all of those things are complete and correct, and they confirm eligibility, then the next then allocation will be revealed. So this is a tool that I think is really exciting, and it can be customized to either be off. So this is, you'll have to click the traditional randomize button, or it can be limited. This will work, but it will be limited to a specific set of users, or it can be used. It can be open to be used by anybody. The next one is one that we had to think about a lot, and whether or not this tool should be available. And it's changing, it's allowing anyone to change allocations within the randomization code. So let me explain what the need is here. So there are rare circumstances in which a patient is randomized and the randomization that is revealed is different than what's in the allocation table. So for example, during downtime. So REDCap is down, or Wi-Fi at your institution's down. Many trials have procedures for how you would continue to conduct a study during downtime, like a coin flip. So if the randomization database is down, then you can flip a coin and notify the coordinating center, and then proceed with enrolling the patient. But now you've got a problem. You've got a patient who's been randomized and enrolled, and their allocation's different than what's in the randomization table on REDCap. And that problem's usually been solved on the analysis side, that we build a line of code in to correct that allocation in the back end. But it creates unease for lots of people that now your study database that might be exported and shared doesn't match truth. So the request was made to allow editing, but at the same time, the randomization sacrosanct, and you can imagine all kinds of misuse for a function like this. So the decision was made to create this tool, but to limit it not to the investigators or even the CCC folks, but to limit it to REDCap administrators. So the people who run the back end of REDCap, but aren't involved in any specific study, and they can, this would only then be available through direct request to them with explanations of why it was needed. Next is connection to API tools. So one thing that became clear as we worked through this process is that when you're doing adaptive randomization, that's one of the most challenging parts of this. So the idea that you want to change the allocation probabilities based on the baseline characteristics of prior patients for minimization randomization, or on the outcomes of prior patients for response adaptive randomization. That's a complicated procedure that requires querying variables, sometimes many variables, from other participants in the REDCap record. And this is something that's going to be customized significantly based on your study. So your study obviously has different covariates that you think are important to balance. It has different outcomes. And it may have different threats, different variables that may be missing, and how they want to deal with missingness may be very different. So it was clear very quickly that this is not something you can hard code into REDCap. But we want to make it easy for people who want to use and build these tools to still have the protections that are built into the REDCap randomization tool. So we've done that by allowing this REDCap randomization tool to connect to API tools, so that you can call an API tool that will then query the REDCap database, get the information that's needed, run a model, and then upload a group assignment, reveal it to the clinicians, and importantly, replace it in the allocation table so that it's there in the study database, in the same study database as the rest of the data elements, and it has the same kind of data protections and metadata that are helpful in cases like this. And then I'll admit, as a clinician and not a biostatistician, I'm getting into my least comfortable area talking about hook functions. But the way I understand it is hook functions are functions that try to make it very easy to know where to link in your API tools. So it's a pre-specified location in the REDCap randomization tool to which external modules can connect. And it calls those external modules at the point when you click on the randomization button and call for the next randomization. And it should increase the ease and flexibility of developing external modules that then could be reused by other people who maybe don't have the sophistication to build their own API tool and run their own server, but might be able to update some metrics in an existing external module. Great. So let's go back to our example trial. So this trial that I talked about as being one that we might think about running in a network like ours, a placebo-controlled dose-finding trial of a novel antiarrhythmic following prolonged cardiac arrest. So can this new REDCap randomization tool support all of these features? Yes, we could embed randomization in a survey and allow any paramedic to access it using a QR code on their phone or on a tablet and truly do a decentralized pragmatic trial in an ambulance. We could choose that for that randomization to either reveal group allocation directly in an unblinded trial or to reveal a randomization number for a blinded trial, and that randomization number might then be linked to a numbered medication pack. We have the ability to change the allocations for future patients and drop an arm at any point in time. We also have the ability to use more sophisticated modeling to do adaptive randomization to balance baseline covariates using an API tool or an external module. And then finally, we still obviously have the ability to do things that don't affect the randomization tool itself, like a Bayesian sequential design. So what are next steps for the REDCap Randomization 2.0 project? It includes ongoing testing. So we're excited that we've already heard about some studies that have been waiting to start and waiting to use this tool. But we hope some people on this phone call will similarly be excited about these tools and will use them and provide feedback to us when they identify further needs or problems with the new software. We're planning to incorporate these tools into the REDCap 21 CFR Part 11 validation framework so that they can be used for FDA registration studies. We're drafting a manuscript on the methods of its development to help with dissemination and increase awareness of these new tools. And then we're developing some metrics to track their uptake and receive feedback. All right, so that's the end of what I was invited here to talk about, which is the new REDCap tool. So now I'm going to turn to opinion, and I'll share some things that I think may be issues that are some of our colleagues think less about, but I think a lot about as a clinician who enrolls patients and runs a coordinating center. So what's the toughest question for adaptive trials? I think it's when to use them. And I think a corollary is why we haven't used adaptive randomization in any of the trials that we've done in our network. And I think some of it comes to trial efficiency. So when I think about trial efficiency, there's at least two axes I think about. There's statistical efficiency, and I'm here presenting today with 4 other biostatisticians, so I think that's probably where they spend a lot of their time thinking about statistical efficiency. I'm a clinician and I run a clinical coordinating center, so I think a lot about operational efficiency. And in this paradigm, I'm going to argue that those two are related, that the information you learned is related both to the number of patients you can enroll for the dollars you have and the information that you get out of those participants. So information learned in this paradigm is the product of those, the area under the curve. And I think there are cases where you can increase statistical efficiency without affecting operational efficiency. So let's go through an example. So let's think about a 100-patient phase 1 trial of a novel infusion for treatment of hypotension, which is planned to enroll at 30 sites. So this is a trial that might look familiar to a lot of patients, and it's a trial in which every patient is going to have lots of contact with a research personnel. So it's going to take a long time to identify these patients, recruit them, and enroll them. And then you're going to spend many hours with the patient as you watch them during the infusion and collect data. And you're also going to be worried if you're collecting 100 patients at a small number of sites. You might be very worried about imbalances in baseline characteristics. So you might be really interested in using adaptive randomization to balance baseline covariates. So what's the effect in a trial like this of doing adaptive randomization to balance baseline characteristics? I would argue that you get that basically for free. So because those research personnel are already spending a lot of time with these patients, they're already going to be collecting that same data, and you're going to spend a lot of time in a phase 1 trial ensuring safety and talking to patients and clinicians before randomization. The added time required to collect 5 or 6 baseline characteristics before you randomize is essentially nothing. So in a case like this, you might increase statistical efficiency by 25% and increase overall efficiency by 25% with no impact whatsoever on operational efficiency. Let's turn now to a different area, maybe an area where it's easier to enroll patients. And I'm going to use here, this is our first case that's not a hypothetical example. This is a trial that we're doing now. So a 2,364 patient comparing the effectiveness of sedative medication during emergency tracheal intubation at 6 sites. So obviously we're enrolling a large number of patients at a small number of sites. What's the benefit of using adaptive randomization to balance baseline characteristics in this study? You may argue that you still have benefit, as we heard about in our first lecture. You may worry that that benefit might be less because the chance of imbalances is smaller when you have a large number of patients, but it still may be present. But there's real challenges. So in this trial is looking at emergency tracheal intubation, where the time from the decision to intubate to the completion of the procedures is 5 minutes. And these procedures are being performed without a research personnel there. So today, as mentioned, we're doing this with envelopes. There are sequentially numbered envelopes in our EDs and ICUs, and clinicians decide to intubate while they're setting up. Someone grabs the envelope, opens it, it tells them ketamine or etomidate. They deliver the intervention and collect information on a one-page data sheet, and they enroll nearly all of their patients into these trials. But if I ask them to capture a number of baseline characteristics, log into a web-based platform, input those characteristics and click a randomization button, even if they were easy to capture characteristics, there's no way they could do that during this brief 5-minute period to identify the patients, enroll them, and randomize them. So I'm confident if I asked in this current trial for them to do adaptive randomization, that I might lose more patients than I gain in efficiency. So I might increase statistical efficiency by 25%, but have the number of patients I'm enrolling and decrease overall efficiency by 40%. This is not to say that any of these tools are good or bad, but I do think that we have to be really thoughtful about what the pros and cons are for every specific study. So when it comes to this hardest question of when to use these tools, that's what the 10 exist for. So Dr. Hanley mentioned that the Hopkins Trial Innovation Network, that center that he leads, as well as the Vanderbilt Trial Innovation Center that I helped to lead along with Chris Lenzel and Gordon Bernard, is really focused on helping investigators answer questions like this. So it's a service that is available, that offers no-cost scientific consults to investigators. We're always excited to help investigators identify and use new tools to increase the efficiency of trials. And we were particularly excited about getting people to use these new REDCap randomization tools, but we're also careful to make sure they're fit for purpose, to only recommend them in times that it's really going to help the study and not just to use the tool for using it. So I think I'll stop there and turn it over for questions.
Great. Thank you so much, John. And a terrific infomercial for the Trial Innovation Network. You're probably going to get an Emmy Award for that one.
I'm trying to give you lots of business.
Okay. I thought that was a stimulating presentation.
Interesting, yes, absolutely.
Questions are beginning to come in. While they're coming in, we will put in the chat in a little while, and at the end, Frank Harrell is writing a commentary about this program, and we'll put the URL in for everybody to look at Frank's thoughts on the program. Guyan?
Absolutely, yes. And so, yes, like as Dan Henley said, we have questions coming in. So the first one is about admin edit tool. Does it automatically generate a log or a metadata record?
It sure as hell should. I'm 99% sure about that, but why don't I take that back to Paul Harrison team and confirm, because you're absolutely right that is a critical feature. I'm confident it does. Every time you log in, access the record, or change any data element of any type, it logs that, and certainly it should be logging changes to an allocation ratio to an allocation, some of the highest risk changes. So let me take that back and confirm it, but I'm 99.9% sure that's the case already.
Great. I'm pretty sure as well, John, that it's part of your CFR Part 11 compliance, which is what the FDA wants to see in terms of who touched the data. Back to you, Guyan.
Yeah. And also for the record, we are as users of and you know, for planning and conducting trials, we're very excited to see the new version of the randomization module for sure. Although I don't think it's here at Hopkins yet, but it's coming. The question is about the comparative effectiveness trial. The just for baseline covariates. I think the David is referring to sedative trial. And did you consider smart design? I think it's linking to the previous presentation on smart design. So what do you think, John?
Yeah, David is a close colleague, and he's one of our partners in the Vanderbilt Trial Innovation Center. And so he's asking the tough questions right out of the gate here. So I think I wish Michael was still on to have a robust discussion. We might turf some of this to the panel discussion at the end, because certainly I think this is an important topic. I think we, in our trial, we have unadjusted models and adjusted models of various types. The question becomes, how do you power the trial? Do you power the trial based on an unadjusted model or based on adjustment? And we, as Michael recommended, we make the most conservative assumption that we should say that we want the trial to be large enough to detect a difference, even if there is no benefit for adjustment. So then the question becomes, what should your primary analysis be? And we, in most of our trials, have continued to use an unadjusted primary and then conducted adjusted secondary analyses. And I'm happy to talk about why that is. And I think a big part is our group, hence our name, the Pragmatic Critical Care Research Group, we're studying questions for commonly used tools that are given to millions of people every year. So compared to other trials, like a phase 1 drug development trial where there's a massive cost to every patient, our per participant cost is very low. Our goal with these trials are to take long-standing questions that have been around for decades. Ketamine and etomidate, that question's been around for 50 years. There have been a number of observational studies that have given conflicting results. There have been a number of small to moderately sized trials that have given conflicting results. So our goal is to do the biggest and most definitive and inarguably true trial ever done that will hopefully settle the issue once and for all. So in our opinion, that for cases like that, where the per participant cost is low, the risk to participants is low, these are the same two drugs they are already getting and the drugs that are being given to millions of people, that we want it to be the simplest and most transparent analysis possible, to be very convincing to clinicians. And that's the reason we've prioritized our unadjusted analysis. I'll say in our 20 trials, we haven't had a single trial where those two weren't concordant, where the unadjusted and adjusted analysis have matched every time. So that might be because we are preferentially being conservative in our sample size estimates and making sure that we're overpowered for our trials. But thankfully, that has not been, there's never been a case where those have been discordant in a way that we regretted. And then finally, sorry, go ahead. So we'll stop there, then we'll get to David's last question.
Well, I think Frank has a comment on.
Yeah.
Go ahead,
We need to continue with the adjustment.
Yeah, I think Jonathan opened up a hornet's nest there. And I will say, I think it's very hard to defend not adjusting in almost any case. And one of the reasons I say that it's an ethical issue, but that's more true when you have like a new drug where you're exposing patients to a possibly hazardous treatment. And if you randomize more patients than you need and expose more to the experimental agent, it could be a big ethical issue. There's discussions about this at FDA, for example. But I think Jonathan has made an assumption that I think a lot of people make, and I want to really question, and that is that something is simple or not. I think the main reason something is simple is because people don't understand it well enough to know how complex it really is. So if you have, let's say mortality is your outcome, and you think of your data as a 2x2 table, the statistical methods that were developed for that, which I think Michael really agree with this, assume that you have the same probability of death for everybody in treatment B. And, we never have that. We have heterogeneity in the outcome. And so if you do a chi-square test, you're assuming everybody in treatment B has a probability P sub B of dying. It's a constant. And so if you think of using a very simplified method that makes it seem simple, you're actually creating something that's hard to interpret. And if you go to time to event analysis, it's more clear. So let's say the males and females have much different prognoses. When you look at the survival distributions for males and females, they may have different modes. And so if you don't adjust for male and female, your survival distribution is a mixture of two distributions. It may even be bimodal. So now you're comparing a bimodal distribution for treatment A versus treatment B. And what that comparison means, which will also violate the proportional hazards assumption in many cases, what that comparison actually means is very complex. So you're making something have a bimodal distribution that if you'd only adjust it for sex, you would have really, really simple distribution. So I just want to push the idea on Jonathan that simplicity is usually hidden complexity.
Frank and I, Frank trained me. I did my master's degree taking Frank's course, so I've been learning from Frank for more than a decade, and you might imagine that we continue to have these discussions in many of our trial innovation center consults. I think that Frank's exactly right that there is no defense for not doing an adjusted analysis, and we do one in every trial. I do think that reporting of trials also has some. There is also, I'll say two things. One is that you're not just reporting a trial, you're trying to convince clinicians. And I think if you have a trial that enrolled the last patient you needed to answer this question in an adjusted model and failed to reach significance in an unadjusted model, I think clinicians will not believe it, especially for something that's already had 8 trials before. So I think that that might be exactly what you want, stopping the trial the moment you hit a p-value of 0.049 in a phase 1 drug discovery trial. And I think that we should have a higher threshold for proving that we've got an effect when we're looking at therapies that millions of patients are getting every year. And the idea that it's unethical to go from a patient 1,000 to patient 1,030 because we didn't adjust, to me, holds no water. Now, the other thing is cost of complexity. So Frank chose a great baseline characteristic. So sex, that's easy to define. I'll say in our consults, it's amazing how many times people come to me proposing that the primary analysis of their trial is going to adjust for something that's actually not a baseline characteristic, that looks like it might be a baseline characteristic, but they're mistaken. So let me give you an example of that. So in airway management trials, we do a lot of trials looking at how you intubate patients. And we get beat up all the time not adjusting for difficulty of intubation, for difficult airway characteristics, for large tongue, small jaw, things like that, small mouth opening. And we ask those in every trial. And the reason we don't adjust for them is they're reported after the procedure. So you'd think, well, large tongue is a baseline characteristic, small mouth opening, that's a baseline characteristic. And ideally, providers would assess them before randomization. And you could force them to do that, but that's not the way most trials do it. They ask afterwards because it's an emergent procedure. They have to intubate the patient emergently, and then they report, do they have large tongue or not? So in our most recent trial, we said, well, they don't have time to interrogate 20 difficult airway characteristics, but why don't we ask them before they randomize on the outside of the envelope, is this going to be easy, moderate, or hard? And we did, and we showed that those two were exactly balanced. Easy was balanced, moderate was balanced, hard was balanced. No difference between groups and no effect modification. The intervention was effective, and was effective in all 3 groups without any effect modification. But on the back end, when the intervention was so effective that people thought, hey, when I use this video scope and intubated patients well, did they have a large tongue? Nah, their tongue wasn't large. And that same patient, if they intubated them with a direct scope, was their tongue large? Oh yeah, that's the reason I failed, is because their tongue was large and their mouth opening was small. So if we tried to adjust for what looked like a baseline characteristic, it would have heavily biased our outcomes. And I will say that I agree with Frank. If we had absolute knowledge of exactly what baseline characteristics were associated with the outcome, and I was guaranteed, I knew that there was no chance that these were ever biased, I might feel more comfortable. But in our space, that has been one of the reasons that we have historically leaned towards unadjusted analyses, is that been the best prognostic factors you want to adjust for most are often biased in their reporting.
I think those are super points. I think it's not so hard to guard against that, but the extreme cases that I have no patience with are studies in which older patients die sooner than younger patients and age is not adjusted for,
Oh.
which is very common. It's not defensible.
Dan, I think you have a comment, right?
Just a quick one. I think in our studies, and that's one of the reasons that Michael was on the presentation group today. And by our studies, I mean ones that Guyan and I and others have done at Hopkins in our main area. We're more on Frank's side because we're not dealing with diseases that have 10 other studies. We're dealing with first of a kind phase 2 and phase 3 studies. But where there's epidemiologic description of true baseline covariates that can't be changed. And I think John made a wonderful point about that. And in those instances, we've found that the efficiency of that Michael talked about is a value, which was the first case that John showed you. And a second reason for using this particular design element is that the interventions were severe. We're talking about doing neurosurgery on a patient and a brain procedure. So I think tailoring the design, which everybody has said in their discussion, and I think we're going to hear more about that from Frank, to the question at hand and the stage at which that study or trial is occurring is very important. I don't know whether you want to say any more about that, Guyan.
I think you described it very, very well. Is that it was definitely a different situation than what John was talking about in terms of reporting and in terms of being a true baseline covariate.
Right. And I guess one other thing I want to say is we need trials in all of these areas, okay? Because as John suggested, there are important questions about the care that we give that remain unanswered that decentralized and large pragmatic trials can answer.
Absolutely.
And there's true value there. And there's true value in smaller disruptive trials that seek to really change the pathway of care. And I think Kelly gave us some nice thoughts on that area. Do we have other questions?
We have one more, so I don't know if we should take it.
Yeah, let's do it. Let's do it. I think there's still plenty of time because we have the transition time before.
I think Frank already answered it by the time we got to it, but basically was a question about the covariate adjustment randomization versus the standard stratified randomization.
Let's hear Frank's answer.
Let's see, am I still unmuted? Yes.
Yes.
Yeah, I think there's something Jonathan may want to say about this because there's some issues of balancing on covariates as you proceed using dynamic or minimization, but other people have questioned whether the balancing of covariates in a dynamic fashion is worth all the trouble. But in my answer, I mentioned that when you do an analysis of covariance, if the treatment has any correlation with any baseline variable, in other words, there's a tiny imbalance, doesn't really amount to much, but that tiny imbalance creates a very slight collinearity between treatment and a baseline covariate, such that when you get the standard error of the treatment effect, it'll be a little bigger. So why not do something that's pretty free and make sure that can't happen so your standard error of the treatment effect is going to be minimum?
John, do you want to comment or?
No, I'll happily admit my ignorance regarding that very tough question, and I'll defer to Frank on that point.
Thank you, Frank.
Well, I'll add one more piece of information, which unfortunately, Guyan and Michael and I haven't published quite yet. We did adaptive minimization in several of our trials. And I think the issue has been brought up. It depends. The ability of adaptive minimization to limit the effect of covariates, baseline covariates, depends upon how you start, when you start the minimization, and the priority you give to the factors that you're minimizing and the sequence that goes on in the minimization of those factors. So in 2 of the trials that Guyan and I have done, it took about 150 to 200 patients to achieve the kind of balance that Frank was talking about. And it was a 500 patient trial. So the second and third factors come into balance a bit later in the trial. So you need a good sized trial, not a super large one, to achieve balance. And 500 subjects starting the covariate adaptation at about 75 subjects. Usually 3 factors have come in line over 500 patients.